When cells divide, they need to replicate their DNA. Telomerase is the molecular machine responsible for extension of chromosome ends after DNA replication. The specialized machine must be maintained at a Goldilocks concentration in dividing cells. In healthy replicating cells, telomerase presides in relatively low abundance due to its high efficiency. Too little telomerase causes telomere truncation and potential loss of genetic information, and the possibility of chromosome end joining by DNA repair proteins. Too much telomerase causes cell immortalization and oncogenesis, or the creation of cancer. Now let's talk about the specifics of DNA replication. The polymerization of deoxynucleotide triphosphates, or DNTPs, is catalyzed by DNA polymerase in a 5' to 3' fashion. The leading strand is capable of synthesizing DNA as a continuous polymer. Whereas the lagging strand performs the synthesis through Okazaki fragments. Unfortunately, this process is perturbed at the ends of chromosomes. When replicating, the lagging strand doesn't have a 3' hydroxyl primer at the chromosome end to continue polymerization, so DNA polymerase can't add the final nucleotides. Since synthesis is not perfectly completed, some genetic information at the end of the DNA strand is lost. This issue happens in every replication cycle and could lead to a lot of lost information and possibly cell death over time. Luckily, nature came up with a clever solution, and that solution is telomerase. This essential complex in the nucleus preserves the telomeres, which are 10 to 15 kilobases of repeated DNA sequence. The shelter-in complex protects the 3' end single-stranded overhang from being recognized by DNA-damaged proteins that would otherwise attempt to repair this apparent DNA break by joining chromosomes together. Exonucleases chew up unprotected DNA, but are unable to when shelter-in is present. Telomerase is a ribonucleoprotein complex with two key catalytic units, the reverse transcriptase called TERT and the telomerase RNA. To begin its critical job, telomerase samples the nuclear environment until it happens upon TPP1 of the shelter-in complex, which is bound to the very end of the telomere. Then the 3' end of telomerase RNA binds the telomere RNA. Once telomerase binds RNA, the telomerase is ready to begin elongation of the telomere. TERT uses its RNA-dependent DNA polymerase function to incorporate DNTPs into the growing telomere DNA chain, using the telomerase RNA as a template. DNA elongation continues until the chromosomal DNA reaches the 5' end of the telomerase RNA template. In order to continue synthesis, the 3' end of the telomerase RNA and the chromosomal DNA strand are displaced, and the telomerase RNA translocates. This cycle of synthesis and ratcheting begins again for another edition of the TTA-GGG telomere repeat. This cycle continues for about 10 to 15 TTA-GGG additions per telomere elongation event. Remember, the right levels of telomerase are important for continued and correct cell division. And that's the end of the chromosome.